A very good afternoon to everyone. Please answer through poll. My audio is clear and my screen is visible. So today we are going to discuss human health and diseases. So health is a complete set of physical, mental, and the social well-being. A person is said to be healthy if it is physically, mentally, and socially fit. And disease is the deviation from the normal physiological condition. For example, when the functioning of the normal or the functioning of the organ or the system of the body is adversely affected, then we call it as disease because there is some deviation, there is some change from the normal physiological conditions. And the biological agent which causes the disease, they are pathogen or parasite. Pathogen which destroy the host body or which destroy the host cell by multiplication, by reproducing. And parasites, they also destroy or they harm the host, but they derive the nutrition, they feed on the food. Now the disease, they can be two types. There's an infectious or the communicable disease, or non-infectious or non-communicable disease. Infectious disease are those diseases which spread from one person to another. Okay, those are known as infectious disease, like common cold. Non-infectious disease are those disease which does not transmit or does not spread from one person to another, like cancer, the genetic disorder. They does not spread from one person to another. Now we are going to discuss different type of diseases like bacterial diseases, viral diseases, protozoan diseases, worm diseases. So first we are going to discuss is the bacterial diseases, and the number one is typhoid. The causative agent or the bacteria which is responsible for this disease is Salmonella typhi, and the mode of nutrition is a fecal-oral transmission. Fecal-oral transmission means through contaminated food and water. This pathogen or the bacteria, it enters into the small intestine of the body. And the symptoms includes constipation, abdominal pain, fever, loss of appetite. And in some severe cases, these bacteria, they can also cause perforation in the intestinal membrane, which is very dangerous. And the diagnosis could be done through the Vidal test. For example, this is a plate. Here's the bacteria or the antigen is present. And if I taken a sample from the infected person, and if I apply the sample, let's say this person has the antibody for the typhoid. The antibody will bind and we will observe the clumping or the agglutination. That means this person is suffering from typhoid or is typhoid positive. Please remember that typhoid is caused by Salmonella typhi, which is the causative agent, and the diagnosis test is Vidal test. Please remember this. It's important. Okay. Next one, there's a lady named as Mary Mellon, and she has been nicknamed as Typhoid Mary because she was a professional cook, and wherever she go and cooked, that person which eat that food, they get infected with the bacteria, the Salmonella typhi, and they get disease typhoid. So this lady, she is the asymptomatic carrier of the typhoid. She's the carrier of the typhoid. Remember this. And the name of the lady is Mary Mallon. Next disease is pneumonia. Pneumonia is caused by the Streptococcus pneumonia, and it's also caused by the Haemophilus influenza. These are the bacteria, and the mode of transmission is the droplet infection or the droplet transmission. That means the transmission through the sneezing or coughing. So these bacteria, they enters into our body and they get attached to the alveoli. When they get attached to the alveoli, they cause infection in the alveoli. And because of the infection, the mucus deposition occurs. And also they cause, they make the pores in the alveoli membrane. And when they make the pores, because of which the extracellular fluid, they enters into the alveoli. So now there's an extracellular fluid is entering the alveoli. Now there's a mucus is also in the alveoli, because of which the surface area gets reduced. So the surface area for the gaseous exchange get reduced, because of which now the concentration of CO2 increases in the blood. That is why the person who is suffering from pneumonia, they have the bluish nail and lips, because there's a high concentration of the CO2 in the blood, and the diagnosis could be done through chest X-ray. Next one is plague. This disease is caused by a bacteria which is Yersinia pestis. This bacteria, it lives in the arthropod or the insect which is a rat flea. And these rat flea, they are either they live in rat or they closely lives with the rat. So when the rat, they bite the humans, when they bite the human, they transfer this bacteria into the humans. And this bacteria that causes a plague disease. They destroy the RBC. You can see the person is having a black patches on the body, is because another name of the plague is also a black death. In this, symptoms includes the breathing constriction, fever, gangrene of toes, finger, tips, nose, etc. Next one are the viral diseases. Viral diseases, the causative agent are viruses. Number one is the smallpox disease, the caused by the variola virus. Next one is the chickenpox, which is caused by the Varicella zoster virus. Common cold, rhinitis. Rhinitis means inflammation of nose. It's caused by the rhinovirus. Mumps is caused by the paramyxovirus. In mumps, you will observe the swelling of the parotid gland. The parotid gland, which are the part of the salivary glands. Hepatitis is caused by hepatitis virus. They affect the liver. Polio caused by poliovirus. And the rabies, they caused by the Lissa virus. First, we are going to discuss in the viral diseases the common cold, which is caused by the rhinovirus. And the mode of transmission is the droplet infection, that is through sneezing and coughing. Symptoms include running nose, red eye, fever, headache, cough, sore throat. When you are having a pain in your throat, it infects the nose and the respiratory passage, but not lungs. And it lasts for 3 to 7 days. Next one is the helminthes diseases. Helminthes diseases are the diseases which are caused by worms, like ascariasis. Ascariasis is caused by the ascaris, which is an intestinal parasite. And this worm, it causes a blockage in the intestine. Mode of transmission is a fecal-oral transmission, that is through contaminated food and water. Symptoms includes internal bleeding, muscular pain, fever, anemia, blockage of the intestinal passage. You can see in the image, these are the ascaris. Next one is filariasis disease or elephantiasis. This is caused by a pathogen or the worm which is filarial worm. That is why the disease is known as filariasis. Or also filarial worm of Wisteria, which is Wisteria bronchopti and Wisteria malayi. Mode of transmission is through a vector which is a female Culex mosquito. Remember this. Okay. So when this mosquito bites, the larva enters into our body, and this larva converts into adult. And when the adult is formed, this adult, they enter into the lymphatic vessels. Okay, so it creates a blockage in the lymphatic vessels, because of which the lymph is diffuse into the tissue and they cause the swelling of the tissue. So in this disease, mostly the lower limbs, they get affected. Okay, you can see this in this image. This is elephantiasis. You can see the swelling of the lower limbs. And please remember the vector name, which is a female Culex mosquito. And the pathogen is Wisteria. Next are the fungal diseases which are caused by the ringworms. And the pathogen could be Microsporum, Trichophyton, Epidermophyton. And the mode of transmission could be through the unhygienic condition or through the wet towel, because you know that the fungi, they require heat and moisture for grow. Like the favorable condition are the warm and the humid places. Symptoms include dry, scaly lesions on the skin, nails, scalp, etc. These all are the symptoms of the disease which are caused by these pathogen, Microsporum, Trichophyton, Epidermophyton. Remember this. Next, we are going to discuss the protozoan disease. This is important. The protozoan disease, which is a malaria caused by the Plasmodium species like Plasmodium vivax, Plasmodium malaria, Plasmodium falciparum. And falciparum, it causes the malignant malaria, which is more dangerous. Okay, remember this. And the vector here are the female Anopheles mosquito. Vector, they are acting as these female Anopheles mosquito, they are acting as a vector because they are carrying this Plasmodium and transferring it to the host. So they are acting as a carrier, they are acting as a vehicle. So that is why they are known as vector. They are called as vector. So the vectors are the female Anopheles mosquito. And this is the life cycle of Plasmodium. This is important. So when this mosquito, it bites the human, now the sporozoids which are present in the saliva of the mosquito enters into the human. Through the blood, these sporozoids, they enter into the liver. Okay, they enter into the liver. Now these sporozoids, they convert into the next stage, which is known as schizont. And these schizont, now they will multiply, they will reproduce, or they will undergo multiple fission and produce merozoite. Now this stage of the Plasmodium, these merozoites, they will start destroying or affecting the hepatocytes of the liver. When the hepatocytes burst, these merozoites are released into the blood. When they are released into the blood, these merozoites, they start reproducing asexually in the RBC. Okay? And they start destroying the hemoglobin. So when they destroy the hemoglobin, they produce a granules called hemozoin granules. And because of these hemozoin granules, the fever occur in humans. Okay. And now these merozoites, they enter into the sexual stage, which is the female gametocyte. They form the female gametocyte and the male gametocyte. But because of the high temperature, because now at this stage, the host or the human, they are having a fever. So the temperature rises, because of which the female gametocyte and the male gametocyte, they cannot form the gamete because they require a low temperature to form the gamete. So these gametocyte, they get arrested. But when the mosquito will bind this infected person and suck the blood, so when the mosquito suck the blood, these gametocyte along with the blood enter into the mosquito gut. So where these gametocytes, they get the favorable environment and they will produce the male and the female gamete. Now the fusion of the gamete occur and the zygote is formed in the gut of the mosquito. Remember, the zygote or the sexual reproduction is taking place in the gut of the mosquito. Okay, and the asexual reproduction is taking place in the human host. Remember this, it's important. When the zygote is formed, this zygote then undergo a motile, form a motile form, which is a ookinete. And this ookinete undergo encystment and form the sporozoid. Now these sporozoid from the gut of the mosquito, they will reach to the salivary glands. So if this mosquito will bite to the healthy person, now again it will release the sporozoid into the blood of the healthy person. And again, the life cycle of the Plasmodium will take place. This is the whole life cycle of Plasmodium. Please remember that the asexual reproduction is taking place in the host and the sexual reproduction is taking place in the gut of the mosquito. So zygote formation is occurring in the gut of the mosquito. Next one is amebiosis. Amebiosis is caused by Entamoeba histolytica. The carrier are the housefly. And here also the mode of transmission is the fecal-oral transmission, that is through contaminated food and water. Symptoms includes constipation, abdominal pain, cramps, stool with excess mucus and blood clot. Because these Entamoeba histolytica, they affect the mucosa or the submucosa. They will start destroying the mucosa layer or the submucosa layer. That is why the mucus, the stool, they have the excess mucus and the blood clot. Next is the immunity. Immunity is the ability of the immune system to fight the disease-causing organism. That is the ability of the immune system to resist the infection or to fight against the pathogen which can cause the disease. So there are two types of immunity. There's an innate immunity or the acquired immunity. Innate immunity, which is present at the time of birth, and it is a non-specific immunity. That means it can resist a large range of the pathogen, or it can block the large range of pathogen. Acquired immunity is the immunity that we get or we acquire during our lifetime. So this acquired immunity we get when we are exposed to the pathogen during our lifetime. And this acquired immunity is highly specific. Okay, so I've already told you that innate immunity is a non-specific defense, which is present at the time of birth, the immunity which is present at the time of birth, and it is a non-specific. It provides 4 barriers. Number 1 is the physical barrier, physiological barrier, cellular barrier, and cytokine barrier. Now let's discuss one by one each barrier. Number 1 is a physical barrier, which is the first line of defense. And this physical barriers, it includes the skin and the mucus coating. Because these physical barrier, their skin and mucus coating, they're acting like a wall. They're preventing the pathogen to directly enter into the blood. So they're acting as a physical barrier. Next one is the physiological barrier. They prevent the microbial growth, and it includes the saliva and the tears, because in saliva and tears, the lysozymes are present, which can degrade or destroy the bacteria. High acidic pH in the stomach, acidic pH in the urine, they also prevent the microbial growth. So these all comes under the physiological barriers. Next one are the cellular barriers. Cellular barrier includes neutrophils, natural killer lymphocytes, macrophages. So neutrophils, they are also known as pus cells, and they constitute 60 to 65% of the WBC. They are the first to reach at the site of the infection or the inflammation, okay, because they're very large in number, because they are present abundant in number. So that means they are the first one to reach the site of infection or the inflammation. Natural killer lymphocytes, these cells, they can recognize the cell with the abnormal protein. We know that on the surface of the cell, the proteins are present, okay? But the cells which are infected or which are hijacked by a pathogen or hijacked by a virus, or the cell like cancerous cells or the tumor cells, they have the abnormal proteins on their surface. So these natural killer cells, they recognize those abnormal protein and causes the cell death, which is apoptosis. Macrophages. So macrophages, they form these pseudopodia like this. They form pseudopodia and they engulf the pathogen. Let's say this is a pathogen. When they engulf the pathogen, now the lysosomes which are present in the macrophages, they get attached with the pathogen and they form a phagolysosome. So the hydrolytic enzyme present in the lysosome, they will destroy this pathogen. Next one is the cytokine barrier. Cytokine, they are the glycoprotein. Example are interferon. This is an infected cell. This cell is infected with the virus. Okay. So these cells, they are producing a cytokine protein, or the example that is interferon. They start producing interferon. And the receptors of the interferon, they are present on the non-infected cells. So when these interferon, they bind with the receptor, they generate a signal and give the signal to the non-infected cell. Now this non-infected cells will secrete some proteins. They will secrete some protein to prevent the multiplication of the virus. So these cytokine protein, they protect the non-infected cells from further viral infection. Next one is the acquired immunity. The immunity that we acquire during our lifetime because of the exposure to the pathogen. And it is pathogen specific. This immunity, it's specific in nature. And there are two types of response are present: primary response and secondary response. Primary response, the response which our body produce when it first encounter the pathogen, and this response, it has a very low intensity. Okay, so when our body encounter first with the pathogen, it produces antibodies and some memory cells. These memory cells, they get stored. So again, if the same pathogen infect the host, or they infect the body, now these memory cells, they can easily recognize this pathogen and they can destroy it. Now the secondary, this is the secondary response, and this secondary response, it has a very high intensity. Okay, so remember, the primary response, it has a low intensity because it is the first encounter of the pathogen. Secondary response, it has a very high intensity. And secondary response is also known as anamnestic. And the primary response is known as the amnestic, and the secondary response is anamnestic. Remember these alternative names. Okay. And these primary and secondary immune responses, they are carried out by the B cells and the T cells. B cells, they produce the antibodies. Okay. And the T cells, they are in the T cells, the T cytotoxic cells are present, T helper cells are present, memory cells are also present. So T helper cell, they help the B cells to produce the antibodies. This is the structure of the antibody. Antibodies consist of two heavy chain and two light chain. This is a light chain. This is a heavy chain. And these light chain and heavy chain, they are joined by the disulfide bond. You can see the disulfide bridges. And the antibody is divided into two fragments. There's FC, which is a constant, and there's FAB, which is the antigen binding. And you can see in the structure, this black portion, they are the variable region. And these variable regions, they are known as paratop. Remember, the paratop is a part of antibody. Okay, remember this. And epitope. Epitope is the part of the antigen. The part of the antigen which binds with the antibody is known as the epitope. And this variable region, it also depends upon the antigen. So that is why we say that antibodies, they're highly specific in nature. And also remember that paratop is consist of variable light region and variable heavy region. This heavy region and light region. This form the paratop, and this paratop, it binds with the epitope of the antigen. This is the site where the antigen binds. So they are the antigen binding sites. And there are different type of antibodies are present. There's IgA, IgG, IgM, IgE, and IgD. IgA is present in a dimer form. Like this. If there's a one antibody, there's another antibody. They are connected with a chain which is known as J chain. So it's present in a dimer form. You will observe these IgA in tears. These are present in tear. They are present in the mother first milk, which is known as colostrum. IgG, they are very small in size. They are the smallest. So they can easily cross the placenta and they can be transferred from the mother to the fetus. IgM, they are the largest. And they are the first to reach at the site of the infection or the inflammation. IgE, they're less than 1%. And the person who is suffering from allergy, they have a more amount of the IgE in their blood. IgD, they are present on the surface of B cell. Remember these points regarding the antibodies. Next one is the type of acquired immune response. Number one is the humoral or the antibody mediated response, and the second one is the cell mediated response. First, we are going to discuss is the humoral or the antibody mediated response. This response is mediated by the antibodies. Look at the image. This is a macrophage, and this macrophage is engulfing the pathogen. So when this macrophage engulfs the pathogen, the lysosomes which are present in the macrophages, they get attached with the pathogen and they form a phagolysosomes. And now the hydrolytic enzymes which are present in the lysosome, they will destroy this pathogen. These are the antigen particles. They have destroyed the pathogen into small particles. These are the antigen particles. Now these antigen particles, they will bind to the MHC receptor. MHC are the major histocompatibility complex. They are like an identity, okay, and they're present on the surface of the cell or the tissues. When this MHC receptor, they bind to these antigen particles, this is a macrophage, right? This whole process is taking place in the same macrophage, okay? Remember. Now these MHC with the antigen particle, they move to the surface of the macrophage. They move to the outer surface of the macrophage like this. Now this macrophage, it's acting as an antigen presenting cell because these are the MHC receptor, and on the this is MHC receptor, they bind to the antigen particles. So this macrophage, they will act as an antigen presenting cell. Now the helper T cell, the receptor present on the helper T cell, they will bind to the antigen and the MHC receptor. And these helper T cells, they will release interferons. And these interferons, they will stimulate the B cell to produce the antibodies. Now the B cells, they will divide. They will produce the plasma cells and the memory cells. Now the plasma cells, they will further undergo multiplication and produce a clone. And these clones, they will produce the antibodies. So this immune response is mediated by the antibody. We call it as humoral antibody mediated response. And the antibodies, they are found in the blood plasma, because humor means fluid. So they're talking about the fluid which is present in the body, which is blood. So the antibody, they're found in the blood plasma. So we call it as humoral antibody-mediated response. Next one is the cell-mediated response. This response is mediated by the T lymphocytes. We know that the T lymphocytes, they have T helper cell, T cytotoxic cells, and the memory cells. These T helper cells, they enhance the immune response. These T helper cells, they stimulate the B cell to produce the more antibodies. So they're enhancing the immune response. These T cytotoxic cells, look at this image. This is a viral infected cell, okay? And you can see on the top or on the outer surface, the MHC proteins are present which are containing or displaying the viral antigen. So these cytotoxic T cells, the receptor present on the cytotoxic T cells, they will recognize these antigen. And they will release some granules. And these granules will cause the cell death or destruct the infected cell. So here you can observe that the cell is directly involved in the immune response. So it is known as a cell-mediated response. And this cell-mediated response is also involved in graft rejection. Graft means organ. So before the organ transplantation, it's very important to match the blood group. It's very important to match the MHC, or the tissue matching should be done. Because if the blood grouping and tissue matching is not done properly, then it then it will lead to graft rejection or the organ rejection. So the person who has undergone transplantation, they are given immunosuppressant. Okay, remember this. Next one is the type of acquired immunity. There can be active immunity or the passive immunity. First, we are going to discuss is the active immunity. It is the immunity in which the antibodies are produced in the host cell. For example, when you are exposed to the pathogen, your body will produce the antibodies against that pathogen. That is known as the active immunity. It could be a natural or it could be artificial. Natural, when you are naturally getting an infection by the microbes and your body is naturally synthesizing the antibodies against that pathogen. But artificial active immunity could be through vaccination, when we inject the dead pathogen or the attenuated pathogen. Attenuated means weak pathogen, or a part of the pathogen or some antigen protein we are injected into the host cells, and now the host cells will produce the antibodies against this pathogen. That is known as the artificial active immunity. Next one is the passive immunity. Passive immunity are when you are injecting the ready-made antibodies into the host cell. Number one is the natural passive immunity. Second one is the artificial passive immunity. Natural passive immunity means the antibody like IgG. I just told you that IgG, they are smallest in size, because of which they can easily pass they can easily cross the placenta, and from the mother they can be transferred to the fetus. So here you can observe the antibodies, they are directly injected into the fetus. Antibodies like IgA, which is present in the colostrum, which is the first mother milk. Artificial passive immunity could be an anti-tetanus serum. Artificial passive immunity, for example, if a person is bitten by a snake, so there's a ready-made anti-venom or the anti-toxins are present against these toxins or the venom, they can be easily injected into the host. Okay, so these are the artificial passive immunity like anti-tetanus serum are present against the tetanus. Immunization is based on the memory of the immune system. There are two types of immunization, active immunization and the passive immunization. Active immunization is through vaccination, and the vaccine could be prepared by taking an antigenic protein or the inactivated pathogen. So when the inactive or the dead pathogen is injected into the body, and our body makes the antibodies against that pathogen. Okay. So if during the actual infection, if that host is actually infected with that pathogen in the future, these the memory cells are also formed, right? The memory cells and the antibodies, they are both formed. So these memory cells, they can easily recognize those pathogen and they can easily destroy that pathogen, which is a secondary response. This is known as active immunization. The different type of vaccines are present, like polio vaccine, hepatitis B vaccine. There's a DPT vaccine. This vaccine is for 3 diseases like diphtheria, pertussis, and tetanus. Vaccines are also produced using a DNA recombinant technology. Example, hepatitis B vaccine produced from yeast. For example, the gene is taken from this hepatitis B virus and it is injected into the yeast cell. And now the yeast cells, they start producing this antigen. This antigen, they can be used as a vaccine. Passive immunization is the direct injection of the preformed antibodies or the antitoxin. For example, the antitoxin against the preformed antibodies against the tetanus or the snake venom, like anti-venom or the anti-tetanus serum. These are the part of the passive immunization because they are the preformed antibodies or the antitoxin against this infection or against this pathogen. And they produce a very quick immune response. Next one is the allergy. It is the exaggerated response of the immune system to some antigen. And the antigen are the allergen. It could be dust, it could be pollen, which are present in the environment. It could be fur of the pets also. And the antibodies which are produced against this allergen is IgE. Please remember, this is the antibodies which is produced during the allergy. And this IgE, they will bind on the surface of mast cell. So when the antigen, they will bind to this IgE, these mast cells, they start releasing histamine and serotonin, which result in allergic reaction like sneezing, watering eyes, running nose, difficulty in breathing, and skin rashes. Treatment could be done like antihistamine should be given, steroid should be given to quickly reduce the symptoms of allergy. And the allergy, they can even cause the asthma, which is a respiratory disease. Next one is autoimmunity. We know that our body can, or our immune cells, they can differentiate between the self and the non-self cell. Okay, but due to some genetic disorder, if the immune cell is not able to differentiate between the self and the non-self, non-self means the foreign cell. If it does not differentiate between the self and the non-self cell and start destroying or start affecting the self cell, then it is known as the autoimmune disorder. And the example is rheumatoid arthritis, where the immune cells, they start affecting the joints. And the Graves' disease, you have also heard. When there's a hypersecretion of the thyroid hormone, then it leads to a Graves' disease, which is also an autoimmune disorder. Next one is the human immune system. It is the system that gives the immunity to the body by recognizing, that means by identifying the pathogen, by responding and remembering the foreign antigen. They play a very important role in allergic reaction, in autoimmune diseases, in transplantation. These human immune system, it includes lymphoid organ, tissue cells, and antibodies. So there are two type of lymphoid organs are present: primary lymphoid organ and secondary lymphoid organ. Primary lymphoid organ, where the maturation, formation, and differentiation of the lymphocytes takes place, the B cells and the T cells. First one is the primary lymphoid organ. The first one is the bone marrow. So the B cells and the T cells, they are formed in the bone marrow, but the maturation of the B cells occurs in the bone marrow, but the maturation of the T cells occur in the thymus. Remember this. The B cell and this T cell. Formation of B cell, bone marrow. Maturation also taken place in bone marrow. T cell formation, bone marrow. But maturation, it occurs in the thymus. And bone marrow and thymus, they both are the primary lymphoid organ. Please remember this point. So thymus, they are the bilobed organ which are seen near the heart, and they are present. They are very large in size during the birth, and as the age increases, their size also reduces. So the immature T lymphocytes, they migrate from the bone marrow and they are matured in the thymus gland. Remember this. Spleen is a secondary lymphoid organ. In the primary lymphoid organ, these B cells and the T cells, they are formed, they are undergoing maturation, but they, from the primary lymphoid organ, then they move to the secondary lymphoid organ where they interact with the antigen. So spleen is a secondary lymphoid organ. It is a bean-shaped organ. It contains lymphocytes and phagocytes. You can see this bean-shaped structure. It is also known as a filtering organ because it trapped the microorganism from the blood, and it also removed the dead RBCs or the worn out RBCs. So it's also known as the graveyard of RBC. It's also the reservoir of RBC in the fetus. Next one is the lymph node. Lymph node also help in the trapping of the microorganism or the antigen. They trap the antigen and they activate the lymphocyte and cause the immune response. Next one is the malt. Malt constitutes 50% of the lymphoid tissue. Remember this. And the malt is mucosa-associated lymphoid tissue. It is located within the mucosal lining of the respiratory, digestive, and urinogenital tract. These are the secondary lymphoid organ like spleen, lymph nodes, tonsil, Peyer's patches. Peyer's patches are present in ileum. Mucosa-associated lymphoid tissue, which constitute 50% of the lymphoid tissue. Appendix, they all are the secondary lymphoid organ. Next one is acquired immunodeficiency syndrome, which is known as AIDS. It is a deficiency of the immune system. In this, the immune system gets suppressed. And it is caused by the human immunodeficiency virus, which is HIV. And the syndromes, this S syndromes means the group of diseases or the group of symptoms. Because when the immune system get weak, now the other pathogens, they start infecting the host cells, and now the other diseases also occur. The AIDS was first reported in America in 1981, and this HIV, it has the single-stranded RNA as a genome. Remember, two strands of single-stranded RNA are present in the HIV, and this is a retrovirus. The virus which can convert retrovirus because they have the reverse transcriptase enzyme, so they can form the DNA from the RNA. They have the reverse transcriptase enzyme. Remember this. It can spread through the sexual contact with the infected person, sharing of the infected needles, transfusion of contaminated blood. and the blood product and from infected mother to her child through placenta. So when the HIV first entered into the macrophages, it hijacked the macrophages, and the reverse transcriptase enzyme, they start synthesizing the or replicating this RNA into the DNA. So when the viral DNA is formed, now this viral DNA get incorporated into the host DNA. So now the virus, they have hijacked the host cell and they start reproducing or multiplying in number. Now these virus, they are start after multiplication, they start infecting or they start destroying the helper T cells. Okay. And because of we know that the helper T cells, they are involved in enhancing the immune response and now these viruses, they are infecting the helper T cells. So okay, so we know that the helper T cells, they are involved in enhancing the immune response, right? So in these viruses, they affect or they destroy the helper T cells. So when the helper T cells number gets reduces, it weakens the immunity. And we also say that the person is having an AIDS when the number of the helper T cell start decreasing. Now, due to the weakened immunity or the weak immune response, now the person might get infected with the mycobacterium, virus, fungi, parasites, and the other pathogen. During this period, the person suffers from fever, diarrhea, and the weight loss. Diagnosis could be done through the ELISA test, which is an enzyme-linked immunosorbent assay. Treatment could be done through antiviral drugs, but these drugs, they are partially effective. They are not completely effective. So these drugs, they can only just increase the life span of the patient or prolong the life of the patient. Next is the cancer. Cancer is the abnormal, uncontrolled growth of the cells. So when the cells, they are abnormally or without any control, they are dividing rapidly or they are keep on dividing and they form a mass of cell which is known as tumor, because the cancer cell, they do not have the contact inhibition property, because when the normal cells, they divide, and when the same cells, they come in contact of each other, they stop their division. This is how they control their growth. But in case of cancer cell, they do not have this contact inhibition property, because of which they keep on dividing and they show uncontrolled growth, which lead to the formation of a tumor. A tumor could be a benign tumor or the malignant tumor. Benign tumor, which are confined to its place, which does not show the metastasis property, which are shown by the malignant tumor. And benign tumor, they cause very little damage. Malignant tumor, they are the most dangerous because they have a metastasis property. Metastasis property, that means they can spread from one part to the another, because these tumor cells, they can slot off from the tumor and they can, through the blood, they can reach to the different parts of the body and they can form a new tumor there. So these malignant tumor, they they exhibit this metastasis property, which is very dangerous. Cancer aging causing agent would be physical agent, chemical agent, and biological agent. Physical, they are the ionizing radiation like X-ray, gamma rays, non-ionizing radiation like UV rays. Chemical agent could be tobacco, smoke, vinyl chloride. Biological agent could be oncogenic viruses, or when the proto-oncogenes get converted into the oncogenes with under certain conditions, they can also lead to cancer. Cancer detection and diagnosis. It can be detection could be done through biopsy. In which a biopsy needle is taken, and with the help of this needle, the tissue is or the sample is taken out from the suspected tissue, and now this tissue is observed under the microscope. The histopathology of this tissue is studied. Imagining techniques, they can be MRI or the 3D scan. You can use the use of antibiotics for the treatment, or the molecular biology techniques, or a chemotherapy can be used to treat the cancer. Next one is the commonly abused drugs. Abused means misuse of something. So there's opioid, there are cannabinoids, and there are coca alkaloids. First is the opioids, which includes the morphine and codeine. So morphine and codeine, they are obtained from the latex of the poppy plant, which are the Papaver somniferum. And the receptor of these opioids, they are present in the central nervous system and the gastrointestinal tract. Morphine, they are generally given by the doctors to the patient who has undergone surgery or who has undergone transplantation, because morphine, they are a very powerful painkillers. Heroin, remember that heroin is a diacetyl morphine. Remember, this is important, okay? That heroin is a diacetyl morphine. It is obtained by the acetylation of the morphine. And it is a white color, odorless, bitter crystalline compound. It is a depressant. It slows down the body function. Cannabinoids, they are obtained from the plant like cannabis sativa. They are generally taken by inhalation and oral injection. They affect the receptors are present in the brain, and they affect the brain and the cardiovascular system. This is the structure of the cannabinoid molecule. And this is a plant, cannabis sativa, which is commonly known as hemp plant. Next one is coca alkaloids, which are obtained from the erythroxylum coca, or the cocaine, which is obtained from the erythroxylum coca. It interferes with the transport of neurotransmitter dopamine. And this cocaine, it stimulates the central nervous system. When the excessive dose of cocaine is taken, then it causes hallucination. Hallucination, it means that it alters the perception of the reality. And please remember that Atropa balladoma and Datura, they are also hallucinogenic plants. Okay. Now let's do the question. Question 1 says, select the incorrect statement with respect to acquired immunity. Number 1 is, an amnestic response is elicited on subsequent encounter with the same pathogen. Second is, amnestic response is due to memory of first encounter. Third is, acquired immunity is non-specific type of defense present at the time of birth. Fourth is, primary response is produced when our body encounters a pathogen for the first time. They're asking about the incorrect statement with respect to acquired immunity. That means the immunity that you have acquired during your lifetime. Very good, Veena. So anamnestic response, they are the secondary response. Anamnestic response, they are the primary response. Primary response is produced when our body encounters a pathogen for the first time. And this response, it has a very low intensity. Because your body is first time encountering this pathogen, right? So your body has a very low immune response or show a very low response in the first. So the intensity of the response is very low. So the fourth is correct, and they're asking about the incorrect statement. So number 1 says that amnestic response is elicited on subsequent encounter with the same pathogen. Yes. Amnestic response is due to the memory of the first encounter. Yes, because amnestic response is the secondary response. Primary response, when your body encounters a pathogen for the first time, then it produces the antibodies and the memory cells. These memory cells, they get stored. So when the same pathogen come in again, these memory cells, they can easily recognize this pathogen and destroy that. This is a secondary response, and this have a very high intensity. Third says the acquired immunity is non-specific type of defense present at the time of birth. No, this is incorrect. Because acquired immunity is specific. And this immunity we acquire during lifetime. But innate immunity, it is non-specific. That means it has a very broad spectrum. And it is present at the time of birth. So third is saying acquired immunity is non-specific. No, it is specific. And it is present at the time of birth. No, it is acquired during our lifetime. So the incorrect statement is 3. Next question says the yellowish fluid colostrum secreted by the mammary gland of mother during the initial days of lactation has abundant antibodies IgA to protect the infant. This type of immunity is called as. Very good, MS Gauri. Very good, Veena. Very good, Sushant. So it's a passive immunity because we are injecting the ready-made or transferring the ready-made antibodies. The IgG, which is because it is smallest, so it can pass the placenta. So IgG and IgA is related to the passive immunity because they're injecting the ready-made antibodies. Active immunity, when the immunity which we get on exposure to the pathogen, when our body form the antibodies against that pathogen, that is the active immunity. Acquired immunity, the immunity which we get during our lifetime. Okay, so the correct option is 1. I hope it is clear to everyone. So already 1, we are winding up the class. Thank you all for your participation.